The association, one moment. The association of individual modules into clusters differs in embryonic and postnatal periods. In embryonic development, the forelimb and hind limb modules cluster together. Sorry. Oh, yes. In embryonic period, the highest separation was observed in hind limb elements. This group of traits had the highest support in the cluster analysis and modularity hypothesis test. Pairwise PLS analysis showed that blocks represented by elements of limbs were highly correlated with each other. The highest degree of correlation was observed for the blocks including the diapodal and autopodal elements of limb. In contrast to the embryonic period, in the postnatal development, the leg module clustered together with the module of head with beak. The hand with primary flight feathers separated from other traits. This was due to distinctive growth of primary flight feathers. If we take only the hand, its growth pattern clustered together with the rest of wing skeletal element. The distinctive pattern of growth had the module of wing variables. In postnatal development, the highest within block integration was in the forelimb traits. PLS analysis showed the highest degree of correlation between the leg module and module of head variables and mass. The synchronization of modules development of head and leg variables in the postnatal period may be due to increased functional load on them that is associated with feeding and the need for chick to rely on hind limbs while moving in the nest. In addition, legs and head are involved in search for food in adults that facilitate the synchronization of patterns of their growth. Also, it may relate to the evolutionary sequence of body plan change in both ancestors. The more distinctive growth of wing variables in the postnatal period is expected because the significant functional divergence of wings and legs in birds and alteration at the beginning of their functioning in ontogenesis implies different patterns of their development. In conclusion, embryonic and postnatal period demonstrates significant continuity both in the dynamics of change in growth and in the clustering of individual modules. The association of individual modules into clusters differs in embryonic and postnatal period, indicating different patterns of integrated trait development. In embryonic development, the forelimb and hind limb modules clustered together. In postnatal development, the hind limb module cluster together with the module including body mass, head, and beak. The strongest modularity was noted for the leg in embryogenesis and the wing in postnatal development. The forelimb and especially the primary feathers have more distinctive growth pattern in the postnatal period. Thank you very much for your attention and listening. I'm ready to answer your question. Any question?
Thank you so much, Oksana. Really interesting talk. It looks like we have a question from Brian in the audience. Brian, would you like to unmute and ask your question? If you prefer, I can ask it on your behalf. I have to let you unmute. Oh gosh, I'm so sorry. Just a minute. You're right. Thank you for catching that. Why can I not just click on the icon? I can't let you unmute because I'm not the actual host. My apologies. Shall I ask your question for you? Is that better? Okay. So Brian had a question. Do you expect similar modularity in other corvids, or are rooks unique in some way, perhaps stronger selection on head size and mass from the brain?
Excuse me, I don't sure I correct. I'm not sure I correct understand your question. Please repeat your question.
Sure. It's also in the chat if reading would be easier. Brian is asking, would other corvids have similar modularity, or are rooks maybe different from other corvids?
From other corvids,
Mm,
maybe in other corvids, these patterns will be different, especially in corvids with different duration of ontogeny. But such study didn't conduct on other species of corvids. Similar study conducted on songs of ash chicks, and patterns of integrated development in this species differ from rook nestlings and embryo. Maybe differences can explain by different duration of nestling periods. Songs or ash chicks have 14th day of nestlings period, and rook nestlings have 30th day of nestlings period, and the primaries in song thrush chicks don't upload significantly as in rook nestlings. I hope I answer your question.
Yes, I think Brian says very interesting points about the period of time in the nest. So thank you. We should probably move on to our next speaker. So Robert, if you'd be prepared to share your slides, and Oksana, if you'd mute yourself, please.
Thank you.
Thank you very much for your talk.
Thank you.
Here. Thanks very much. And thanks to the moderators. Is the audio visual okay so far for me?
Yep, it's fine on my end.
Great. Well,
Thanks.
thanks everybody. I'm really pleased to be able to give this talk, and I'm quite terrified, frankly, to give this talk because it's a controversial one, and I refer to controversial topics, and I want to sort of get that out of the way from the get-go. So, for example, I'm going to make some allusions or refer quite directly, actually, to interpretations of Ronald Fisher's 1930 book, The Genetical Theory of Natural Selection. You will recall in the summer of 2020 that evoked quite a firestorm. Part of that was, or most of that was, related to claims, particularly in the book that's on my title slide here, that Fisher was a white supremacist, and that I'm going to refer to his views on population level differences among humans, also known as race. And all of this is really, really controversial stuff. So again, I'm grateful for the fact that you guys are here and willing to hear me out. Okay, now multi-level selection, group selection, the term I use, which, group selection is part of this grand scheme of multi-level selection. This too is controversial, although not quite in the same way as the summer 2020 Fisher Must Fall movement. Some of you may have enjoyed, as I did, this wonderful workshop we had on Wednesday on multi-level selection. And all that means, and I'm assuming for this audience, this doesn't need too much explanation, but all it means is that the levels of selection that on which natural selection works, like the individual, for example, or the selfish gene, right? Those are two among quite a few different levels on which selection can act. And then it can do so simultaneously. So we can have selection acting on chromosomes or gametes at the sub-individual level and on groups, you know, picture Manhattan, you get the idea, I hope. And a lot of what I'm saying is from a textbook that I really enjoyed a lot, Okasha 2006. And, you know, he's standing on the shoulders of people who came before him, like David Slodmelson and Elliot Sober, among Bill Hamilton, among many others. And so the kind of multi-level selection that I want to draw your attention to for most of this talk is something called MLS1, multi-level selection 1. And this is, I think, the first authors to use this particular phrase, multi-level selection 1, were Demuth and Heisler in 1988. And what they're getting at in that case is very similar to frequency-dependent selection. And I'll explain why I think it's not quite the same in a moment. But before I do, I want to contrast that with multi-level selection 2, which I'm not really going to talk about in any detail. But this is a somewhat stronger claim about group selection at the level above an individual or other multi-levels in the hierarchy, that you can have high-level units in the selective hierarchy that have a degree of heritability. So, for example, Jablonski in 1987 talked about Cretaceous bivalves having heritable geographic ranges at the species level. So multi-level selection 1 is, like I said, similar to frequency-dependent selection. And some of you will, maybe you're fans of John Maynard Smith. He was quite critical of group selection. And in his 1998 review of Sober and Wilson's book, Unto Others, he basically said, well, why do we have to come up with this extra bit of jargon? I don't know if he actually used that term, but he didn't phrase it very nicely. He said, well, it's just the same thing as frequency-dependent selection. And I'm sympathetic towards Sober and Wilson and Okasha, and saying that it is different in the sense that what multi-level selection 1 is doing, like frequency dependence, is generating population structure for the particles, right? In this case, we usually refer to genes in that case. And that population structure affects fitnesses. That's no different than frequency dependent selection. But the difference is the fact that multi-level selection 1, group selection, can produce sustained evolutionary consequences insofar as they reappear, these population structures conveyed by groups reappear over many generations. So that can yield sustained evolutionary consequences. And I think it's worth a brief mention, this great example that Sober and Wilson discuss in their 1998 book, which is in turn based on some research by John Muir in the 90s, that if a poultry farmer selects the most productive hen in a given farm, disaster will result, because these hens are laying eggs in a social context. You have to select at a higher level. You have to select groups of hens, not individual hens. And this is the kind of population structure that has an impact on individual fitness. And I find that quite intuitive. Okay, now what does this have to do with Ronald Fisher of the genetical theory? The last 5 chapters of the genetical theory, these are the controversial ones, chapters 8 through 12. I want to propose to you that he was basically applying multi-level selection 1 to explain the rise and the fall of human civilization. So here's that same quote from Okasha, 2006, generating population structure for the particles, which in turn, according to Fisher, has sustained evolutionary consequences. And in Fisher's case, he actually wrote that these social aspects of high-level units of selection are much more powerful than the direct selection of individuals. So I'm going to go into that a little more momentarily. But before I do, I'm obliged to discuss some of the controversy that I'm sure most of you, probably all of you, remember from the summer of 2020, such as this article by a retired historian from my university who had a rather emotive title, R.A. Fisher and the Science of Hatred. This was an article, popular article in the New Statesman from July 2020. And basically what Evans did in that article, and he was one of many, repeated a claim that first showed up in a blog in 2016. And that blog claimed that Fisher 1930 wrote, that Fisher in the genetical theory wrote, and I quote, that civilizations fail because people of low genetic value, read black and brown people, have more children, right? And that bit between the square brackets is not That was in the original quote that was repeated verbatim by Evans, among others. So, for example, this showed up in the BBC, who wrote Fisher was known for arguing people were divided into genetically inferior and superior groups along racial lines. Now, one of the things that I want to convey to you is that in the genetical theory, Fisher 1930, he did no such thing. It's simply not true. And in fact, there are some quite nuanced passages from the genetical theory that I'd like to share with you. So, for example, in chapter 11 of genetical theory, Fisher 1930, he's actually arguing against a real white supremacist, this French guy by the name of Gobineau. Some of you may know this. So Gobineau was this French aristocrat who thought that the only reason why the North Africans or the Ganges people of the Ganges River Valley or East Asia, the only reason why humanity can come up with monumental civilization is because white people from Europe go out to various corners of the earth and, I guess, teach the locals how to do it. And of course, Fisher disagreed with that. Fisher said on page 239, and he did it in his typical kind of roundabout way, so he's not really obvious. You kind of have to read it carefully, but he's quite clear, I think, apart from Gobineau's assumption of the European origin of the rulers, the historical importance of such cases, meaning monumental civilization, cannot be doubted. That means that he did doubt Gobineau's assumption of the European origin of rulers. And on page 176, chapter 8, he's much more explicit. The experiment of becoming civilized has, in fact, been performed repeatedly by peoples of very different races. Okay, now, this is all part of Fisher's larger corpus, right? So what I just did was cite a few pages from one book that he wrote. I think it's probably his most important book. Virtually all of Fisher's private correspondence is available on the web these days to look through. And if you do that, he definitely said some pretty egregious things that no one listening to this talk, I certainly would not endorse. So, for example, he believed that traits such as heroism, bravery, work ethic, intelligence, traits that clearly have a major social component to them, were heritable to some degree. I mean, you know, these are empirical questions. We could argue about the genetic component to social behavior. I mean, there clearly is one. Everything has some component of phenotype, of environment and genetics. But he was rather simplistic, in my opinion. He also seemed to have believed incorrectly that some major human ethnic groups were real biological units. You know, writing as he was, at least in the lead up to 1930, in the first decades of the 20th century. So we know more about the invasive African species of Homo sapiens that he did. In any event, I'm not trying to apologize for everything that Fisher wrote, but I do want people to know what he actually wrote in the 1930 book. Okay, now what is this multi-level selection 1 group selection mechanism to which I'm referring? I want to give you a little more detail on that point. His group selection mechanism was the disjunction, sometimes the junction, of socioeconomic class with fertility. So he talked both about the rise of civilization and the fall of civilization in these last 5 chapters. It wasn't just about the fall of civilization. So, for example, in regards to the rise of civilization, he's saying that if society was derived generation after generation predominantly from its more successful members, doesn't matter where you are in the world, doesn't matter what the ethnicity of the human beings in question is, he's saying that we can expect civilization to rise. The fall of civilization is, of course, the contrary. That occurs when classes of persons who are prosperous and socially successful are, on the whole, the biological failures. And again, assuming quite a substantial genetic component to not just social behavior, but also fertility. Okay, now, to say the least, Fisher is not generally recognized as having contributed to this concept of multi-level selection. And in fact, he's usually portrayed along the lines of sort of the father of genic selection, the father of the gene's eye view. And why is this? Well, there are a few reasons that I wanted to end with. First of all, I think we're kind of afraid. I mean, my own experience in going through this process, it's kind of scary to read the last 5 chapters in the genetical theory. I mean, not scary to read them, but to talk about them with our colleagues. And Alan Graffin back in 2003 had a really nice quote that I wanted to read to you really quickly. There is still so much to emerge from the initial chapters of Fisher 1930, so the ones that weren't about humanity, that it would be surprising if a close study of the remaining largely neglected chapters did not yield important ideas. And I think Graffin's getting at that same point. We're also used to thinking in terms of Fisher versus Wright. When we teach evolutionary biology, we, within the first 2 hours of lectures, Fisher versus Wright eventually comes up, and Sewell Wright's adaptive landscape is all about groups with small effective population sizes that can let genetic drift take over from selection and thereby cross a fitness valley. And Fisher hated this idea famously. And so we kind of—
About 2 minutes left, Robert, sorry.
Very good, thank you. We're kind of used to thinking Wright is the guy on the group side and Fisher is the guy on the gene side. And that's not quite accurate. And there are lots of authors, a few of them I've cited here, Esposito, Edwards, Frank, who, when you read Fisher and Wright carefully, you can figure this out. But the pedagogy of teaching population genetics doesn't really lend itself to recognizing Fisher's contributions to groups. And then the final slide I have is, of course, that figuring out what Fisher 1930 meant is difficult, famously so. And I want to end with these 2 quotes on page 280 in Fisher, in the second edition of the genetical theory. He writes about the unimportance of higher level selection, and he writes, "It may be doubted if it would be possible to point to any such character with the possible exception of sexuality itself, which could be interpreted as evolved for, a high level advantage rather than individual." That's quite clearly against group selection, but maybe he's talking about this multi-level selection too, right? Whereas on page 245, he seems to say at first glance the opposite. He, as I quoted already, that this population structure that can reoccur over many generations is an agency an agency that is much more powerful than the direct selection of individuals. And I think he's referring to MLS1 in that case. And ladies and gentlemen, thanks so much for your attention. I'd be happy to take some questions.
Thanks very much, Robert, for your talk. Does anyone in the audience have any questions they'd like to ask? Laura, perhaps you can watch the Q&A and see if there's anyone that needs to be unmuted. Do feel free to raise your hand if you have any questions, of course.
Nothing in the Q&A so far.
Okay. I actually have one question I'd like to ask.
Sure.
So obviously, over time, civilizations do change. And for example, class structures can change as well. Do you think that might have something to do with this sort of recurrence of repeated episodes that Fisher was talking about?
I think so. I mean, if I understand correctly, that certainly lends itself, as far as I can tell, to population structure. So yes, is a short answer to your question. I mean, he probably did get wrong some of the really heritable, he was very much on looking for the Mendelian patterns of this or that trait, among other things, and leaning towards a genetic basis for, he discovered that hammer of the Mendelian pattern, and everything looked like a nail as far as he was concerned. So yes, no, that's the short answer to your question is yes. I think that sounds right to me.
Thank you. Any other questions? About a little bit less than a minute. Probably should move on, actually. Thank you very much for your talk, Robert. Really appreciate it. Our next talk is by Tan Pham. Tom, would you be willing to start sharing your slides, please? We can see your slides. They're not yet in slideshow mode, just so you know.
Ooh.
Okay, can you see it now?
Yes, that looks wonderful. Thank you very much. Please feel free to begin.
Hi everyone, thank you all for coming. And then today I would like to just present briefly result for genome assembly of a local adaptation butterfly, Melitida. My name is Tân. I'm from Vietnam and I doing the PhD in Poland now. So as the first one, why we need a good sequencing genome? Because like it's supposed to support for evolutionary biology to understand about the local adaptation, and also to support for study of the genome evolution. and also it can help to understand about the biodiversity and the conservation. So briefly about my species, my working species is like Meridillida is very common species. They have very wide distribution range, and you can see here this is the distribution map of my target species. From it's distributed from Africa to Asia, Australia, and Oceanic country. And my species or my common species also like distribute in both two habitats, open and closed habitats, very congested habitats. And more interestingly, that this species have the seasonal polymorphism. In the dry season form, you can see they have the tail and the tip here, but like in the wet season form, they don't have the tail here. But like you can see, the eye spot is present in the wet season form, but like not present in dry season form. Moreover, this species has the polymorphism in the color wing button. In I mentioned here is like in the dry season form, every single individual have like different wing button color. So my objective of my research is like to provide a good reference genome. The second one is like to identify the genes in the genome, and the last one is to identify the genome arrangement with the close related species such as Mycalessis from the same is from the same choice. And my method is like I am going to use one PacBio risk and one nanopore and the for sequence of RNA sequence. Here is byline of my work from here is my byline. And I just showing the result the primary result because like I didn't get the data from nanopore and RNA read yet. Here you can see this species have quite medium size of genome, and they have very high in heterozygosity, nearly 2%. And after the assembly, we found that like the genome size is like more than 500 megabytes, and we found like 44 context in the genome for primary genome, and this one like we using the Busco to compare with other Lepidoptera species. We found like the community need is like more than 98%, and missing is 1.2%. And I am comparing this species with another species from the same subfamily. Have the diversity is like between the diversity between two species is like 54.4 million years ago, and we can see that like it's very the genome of two species is very highly co-condensed. And in some ways that like we found the genome size of this species the medium size 500 megabytes with the high in heterozygosity, and it's quite similar to the biciclid aninana. And my future plan is like I am going to answer two more questions. The first one is the next one is like the first one is working with like how metadata spread across its range. I working with the biogeography and demography, And the second question after this one is like, how has metadata repeatedly adapt to the new habitats? Uh, so I use the genetic differentiation and analyze about the selective speed. Thank you for the question. Yes, I would like to thank my grant supporting grant and my people support for my work. Yeah, that's all.
Wonderful. Thanks very much, Tan. Does anyone have any questions for Tan over the talk? It's really cool to see a reference genome on another butterfly species.
Yeah.
I actually have a question, and forgive me if you did cover this in the talk already. So melanitis has the seasonally polymorphic forms, right? Do you
Yes.
know, are any of the genes that are known to be involved in coloration in other butterflies also found in melanitis, like cortex or optics?
We didn't found any other species have the season have the polymorphism in dry
Right.
season form like in this species.
Mhm.
We are like in the future I will try to identify which gene responsible for the polymorphism in this one, but we didn't have the data yet.
Totally fair. Thank you very much. I was curious, so I'm glad to hear that's perhaps in the
Yeah.
future. Are there other questions from the audience for Tom? Any questions, any raised hands or Q&As? No more questions. Cool talk though, really great to see. Thank you so much, Tom, for your contribution. Remember, folks, if you think of a question later, feel free to reach out to any of the speakers for the session, and I'm sure they'd be happy to entertain questions from you. We technically actually finish at 45 past the hour. So if people want to stay and continue hanging out, feel free. I will keep the room open, or rather ask Laura to keep the room open until that time in case there are any questions for any of the speakers. And I believe we'll be starting up again in approximately just about a little under 2 hours.
Hi Alex, are you there? I just have a question now.
You have a question for who?
Alex, for Alex, the previous presenter.
Oh, for Robert, the previous presenter.
Robert. Oh yeah.
Okay.
Yeah.
I'm here.
Yeah. Thank you for the presentation. I don't have the question during the talk, but like now I just want to ask like because I didn't read about the this this book about the genetical theory, but like I just have a assumption that like because Fisher working with the human, right? He mentioned about the human, but like I'm thinking because my species have very wide distribution and my species don't have the social like social work, right? Like do not like don't have thinking, so I can test for this kind of thing. I think it's possible for me to test.
Yeah, for sure. I mean, if the workshop on Wednesday, it should all, I don't know if it's all online yet. It may be available virtually for the participants of this conference. I think it all is going to be on YouTube eventually, but there's huge amounts of research. If I understand your question correctly, for a group, you know, these multi-level selection questions are hugely important. And I think, yeah, I mean, I'm not really a good source for that because I sort of stumbled into this topic for reasons that if you want, I can share with you, but it's not, you know, I'm not a multi-level selectionist, I guess, per se in my day job. So I don't know. That's not much of an answer to your question, I know, other than referring to the workshop on Wednesday, which hopefully is accessible to everybody via the online videos and stuff.
Yeah, thank you.
Of course, if it's not accessible, you could always reach out. Oh, Alex is telling us that it is in fact available on the Zoom platform, but I'm sure you could also reach out to the workshop organizers and ask them for any feedback or advice on how to apply it to your system. Anyone else have anything they'd like to ask any of the speakers? Otherwise, I think we'll keep the room open for another 4 minutes, and then I'll ask Laura to close the room down and give everyone a break. To let you know, Orlando, in case you didn't see the chat, the next talk has unfortunately been withdrawn. So if you're hoping to attend it, unfortunately, it will not be shown. Thanks, Brian, for the reminder.
You want to close the session just to limit any confusion?
Yeah, I think that's a good shout. I guess stop recording and then close the session.
Yep. Okay.
Thanks very much for the help.